Uh, Good morning, all, and welcome to the first series of the lecture series that we have started, the MycoAsia and the CBEC together, wherein the first initiation has been taken up by Dr. Belle Damodara Shenoy. Uh, the idea amongst the concept is together between MycoAsia and thing. Those who are new to the thing, I'll just brief about MycoAsia, which has been a journal since 2019, e-journal, and it is having a very high standards and ethics that has been followed because I have been reviewing some of the manuscripts, so it has been following a very good ethics and standards for the publication that was lacking in most of the journals in the era of predatory journals and in the time of so many of the UGC care journals. It is maintaining a good ethics and kind of thing. So this is a journal wherein you can submit your manuscripts related to fungi, and it has an average processing time of 45 days from the date of submission up to the publication, and it is a uh, all your papers are reviewed through two or more reviewers, and a proper comments are taken, and then improvement is done, and it has been published. Uh, it has its website, that is MycoAsia. I'll just try to brief you some of the initiative that has been taken by the MycoAsia. That is, one is the fungi ID that you can see on the right hand side. It is a very good initiative. It's also on the WhatsApp group wherein you can submit your photographs, mostly macrofungi. People submit, but even your microfungi you can submit it to know at least a basic idea of the fungi. Those who are new, the students or young faculties, to get an basic idea wherein you can narrow down to look into further detailed identification. The website also shows about the submit the manuscript procedure, the editorial board, and all the previously published articles, as well as it has started a new journal of Myco India also. All the events that take place through MycoAsia are also listed there in events, where it has got the YouTube channel also, where you can listen to the previous events and the talks also. CBEC, it's a Center for Biodiversity Exploration Conservation. It is an NGO registered under the Society Act, and it has been working on the fungi in the past 7-8 years, wherein they are also working on the quality research, try to do some good work on fungi, fungal taxonomy and ecology, basically. We also conduct the trainings and internships and dissertations, and some research work also we are conducting at present. We are focusing on various habitats of India, and especially in Madhya Pradesh. Its laboratory is situated in Bhopal, Madhya Pradesh. Coming to today's speaker, Dr. Shenoy, I think I do not have to introduce him, but I'll just say some words that he's at present working as a principal scientist at CSIR, National Institute of Oceanography, Visakhapatnam Center. Earlier, he has also worked at MTech Chandigarh and NIO Goa. He has more than 45 international papers, and he's also a managing editor of the MycoAsia journal. He's also on the advisory board of Amity Institute of Oceanography and Atmospheric Sciences, and he has vast experience, I tell you, and he has been he has taken an initiative to propagate mycology, as well as to the students up to the young faculties. He is very active in social media, answering your queries, and he's very popular. He also intends to meet young mycologists wherever, in whichever city he visits. He make a point that he meets them, try to understand what they are thinking, try to discuss on the subject, kind of thing. So we hardly welcome Dr. Shenoy, and I hand over the platform to Dr. Shenoy to please go ahead with his talk. Thank you, Dr. Shenoy. Please go ahead. Please.
Good morning, Dr. Rohit. Sharma.
Good morning.
I hope I'm audible and visible.
Yes.
First of all, thank you very much for your kind and generous words for me. I hope I can do the justice to those words you have mentioned for me. Secondly, I would like to thank you for collaborating with MycoAsia Journal of Modern Fungi and initiate this lecture series on modern mycology lecture series. I'm sure it will be full of diverse people because you are involved now in this lecture series. You will be contacting your connections, your experts, your friends, and you would be recommending and we would be hosting them for MycoAsia-CBEC Modern Mycology lecture series. And importantly, I would like to thank all the participants today for joining in. And thank you very much, for showing your love and support, and not only love, like continuous support and continuous love for MycoAsia and recently for Myco India. So in today's presentation, I'll be sharing my experiences as a fungal taxonomist. About me, Dr. Rohit has already told, but let me give some more ideas about my educational background. How did I get into fungal taxonomy? The personal touch probably will have some effect to the story I'm going to tell. Let me start sharing the slides. Rohit, is my first slide visible to you? And the ladies and gentlemen in the audience, can you please let me know whether my slide is visible?
Visible.
Yes.
Yes, sir.
Yes, visible, sir.
Great. Okay, fine. I have titled.
Yes, sir. Visible, sir.
Great. Thank you so much for your responses. Let me just mute all of you for some time, maybe for the next 45 minutes, so that there will be uninterrupted flow of monologue from my side. Then we will have a dialogue, maybe after 45 minutes. I hope it's fine with you guys. Okay, sorry for all those interruption in between, but I have, as I was telling, I have titled today's talk as how to identify a fungal species. So based on this title, I can ask myself 4 simple questions. First one, fungi. What are fungi? Second one would be, what is a species? Third one would be, what is identification? The fourth one, important one, how to identify a fungal species. This is a little bit complicated. It's technically complicated. If you, as I am going to talk about it in next slides, you will understand why it has become a little bit complicated. But it is important to identify a fungal species. That you will understand as I move towards this presentation. Ladies and gentlemen, I'm Belle Damodara Shenoy. I did my MSc from Mangalore University in Karnataka state of India. I did my MSc in biosciences, where Professor Sridhar was my teacher. After my MSc, he connected me with Professor Kevin Hyde, who was working at the University of Hong Kong then. I joined for my PhD at the University of Hong Kong under Professor Kevin Hyde and Dr. Rajesh Jeevan in 2003, and I worked on using molecular data to connect anomorphs with the possible teleomorphic taxa. Then after my PhD, I joined Dr. Keith Seifert at Canada as a postdoctoral fellow, maybe for a brief period. For a brief period of time of 4 months. Then because of my mentor and advisor in India, I could get into a good job in MTCC, CSIR, IMTech Chandigarh as a fungal taxonomist. I spent 5 years there as a fungal taxonomist. During that time, my PhD students, Dr. Gunjan Sharma, Dr. Yamini Agarwal joined me. Gunjan worked on qualitative species. Yamini worked on entomopathogens. Then after that, in 2013, I moved to CSIR National Institute of Oceanography. I am at CSIR National Institute of Oceanography Regional Centers, Visakhapatnam, which is on the east coast of India. I am a principal scientist there. As many of you know, I am one of the co-founders of MycoAsia Journal of Modern Mycology. Recently, we have started a new journal, Journal of Indian Fungi, where Professor Vandikare Ramaya Sridhar, my MSc teacher, is the editorial board. This is my email address, shenoy@gmail.com. You can always contact me on my WhatsApp. You are free to connect with me on LinkedIn also. So this is the logo of my organization, CSIR National Institute of Oceanography. It is based in Goa. The headquarters is in Goa. As I told you, one regional center is at Visakhapatnam. Other two are there on the west coast of India, Mumbai and Kochi. And we do multidisciplinary research from physics, ranging from physics, chemistry, geology, biology, ocean engineering, instrumentation of oceans. Actually, you are welcome to visit our website, www.nio.org. So what is the plan for today? The plan is that first I will give you some basic understanding about fungi. Just to get you into the topic, actually. I don't want directly get into the complicated topics of fungal taxonomy, as there are many beginners in taxonomy and beginners in mycology. For this benefit of such people in the audience, I'll be starting with simple things. What are fungi? Some interesting facts about fungi. Then I will move towards fungal taxonomy. What is taxonomy? What is identification? Then I will give you some ideas what I did during my PhD work. Okay, then I will talk about Gunjan's work on Polytrichum. Then I'll come to the core issue of what constitutes a fungal species, how mycologists have been recognizing fungal species. Then finally, I am going to share some of the guidelines which have already been published about those guidelines that can be followed when you describe a novel fungal species. And at the end of the question and session, you're free to ask me questions. There are a couple of surprises. If you stay till the end of this program, we will be sharing a feedback form. If you submit the feedback form, Dr. Roy Sharma will issue certificate of participation in the name of CBEC. And second thing is that we will be selecting a couple of participants who are active on the chat box, asking questions, in the chat box. Maybe later in the question and session answer. So you will select based on their activity, their enthusiasm, their kind of questions they want to ask. So 2 lucky people will get Amazon vouchers at the end of today. Okay. A simple question: how do we identify people? Probably you can identify one person based on the face. Or you look like your father, you look like your mother, or you look like someone. So that means to identify someone, we need to have a priori knowledge, a previous knowledge we should have about the context. The government also identifies people. In case of India, we have other system, UID, Universal Identification System. In that system also, the biometric data and other data of people been already collected. There is a priori knowledge, priori information is done. Based on the priori information, identification is done. When you input your Aadhaar card, it will tell who are you. If the record is not there for you, it will say the information is not there, record not found. Then you have to go to nearest Aadhaar Kendra and give your details, your fingerprints, other details to be enrolled part of the database. So what I'm trying to say is priori information is very important in case of identification. And identification is a binary process. It will tell whether that particular entity, individual, or someone or something is identified when we compare against a known information or a database. Okay, that's the binary. It's yes or no process. In case of fungi, how do we identify fungi? Simply, if you are not a mycologist, probably you look at say, okay, in this picture, I can see some mushrooms. So I know that mushrooms are fungi. So based on morphology, external appearance, you can identify. And this knowledge of identifying organism, especially identifying fungi, is important. That you can realize if you go to Google search. You type how to identify fungi. People have been asking different interesting questions on Google. People want to know how to identify fungi under microscope. They want to know how to identify fungi in lab. They want to know again how to identify fungi on other plates. And then some people want to know how to identify fungi in the United Kingdom. And some smart people want to know how to identify fungi species. Some people want to know how to identify fungi morphologically. And in some medical fields, probably people from medical field, probably they are interested in knowing how to identify fungi cell or fungal cells, and how to identify fungi from spores. Okay, can we identify a fungal species, assuming that you know the concept of fungal species, based on the appearance of fungal spores? Is it that simple? That you'll come to know soon. As I told you, this question is very important, how to identify a fungal species. People have been asking such questions on Google, how to identify fungi species, how to identify mushroom species, how to identify yeast species, and how to identify fungi, and how many species of fungi have been identified. That's an important question too, actually. So before I move on to fungal taxonomy part, I want to go through some beautiful slides about 10 interesting facts about fungi. So I believe, and I strongly believe, fungi are beautiful. They are beautiful. They are beautiful, actually. Oh, probably you may not say the same thing here. Maybe you disagree with me. No, here this fungi or this fungus is not beautiful. It is actually spoiling of food. But if you remove the human biases, if you just look it as a somebody who appreciates beauty, probably in this particular case also, fungi is beautiful. Okay, as we move towards there are some more pictures of fungi here. Here you have some fungi on culture medium, actually. I find them beautiful. These are like imaginary photos from canva.com, or artistic representation of fungal spores and I have shown these pictures as I believe, as many of us believe, that science is about evidence, but science is also more about imagination. If you can let people imagine the beauty of fungi through these slides, probably we can get them into the field of mycology. And as I have been telling in many of my presentations, that the largest living organism in this world is not what you think it is. You already know that largest living organism on this earth is a fungus. It's a mushroom. It's the name of the that fungus is honey mushroom, Armillaria ostoe, and it is found in the United States. Why it is the largest? It's the largest by area because the mycelial network beneath that mushroom bodies, they cover around 965 hectares, actually. And it seems this particular fungus mushroom and the mycelial network weighs around 400 tons. And it is not me telling or this website telling. This is the Guinness World Record, actually. Probably it is a credible information. As you know, life has been divided into 3 domains: eukarya, bacteria, archaea, and our fungi, animals, plants belong to eukarya. Let me clarify that fungi are not plants, even though we have been studying fungi in the department of botany or in the botany, but fungi are not plants. Are they animals? No, fungi are not animals. Fungi are fungi because they have a separate kingdom. If you see this tree of life, here you have a separate clade for kingdom fungi, and there is a higher level phylogenetic classification of the fungi, which is based on multi-gene sequence data and polyphasic data, which confirms the monophyletic clade of fungi. We have included the Macrosporidia as part of monophyletic kingdom of fungi. Going to the second point about the interesting facts about fungi. Fungi can be microscopic, as you can see here. I mean, you cannot see here, you can just see a green patch here. But if you put it under microscope on the culture media, you might see like that. Then if you use microscopes, you can see the detailed structures. Okay. But not all fungi are microscopic. Many are macroscopic, like in case of mushroom, they are macroscopic. And when they are macroscopic, they are beautiful and they have greater impact actually. And if you talk, I was talking about fungi, plants, animals being part of eukarya. Then who came first to the land? Whether plants came first or the mushrooms or fungi came first. According to some data, long before trees overtook the land, earth was covered by giant mushrooms, and the giant mushrooms were as tall as 24 feet. So here is a pictorial representation of those giant mushrooms, and they were dominating the ancient world, but they disappeared because of many reasons actually. Today we see lots of plants and trees, but not giant mushrooms. Another important question is how many fungi are there? I mean, there is very difficult to describe individuality in fungi, but you can describe in terms of species. Okay. I would rephrase this question as how many fungal species are there? According to one estimate, there could be at least 15 lakh fungal species on this earth. So that means so far we have described only 2 lakhs fungal species. The remaining 13 lakhs we have to discover and describe in a proper taxonomic way. That taxonomic way I'm going to describe in the latest part of my presentation. So there are some, see, these are the edible mushrooms, popular ones. These are the 18 edible mushrooms from the world. That include cremini, morel, shiitake, lion's mane, portobello, chanterelle. So, but not all mushrooms are not edible. As they say, you can eat some mushroom probably only for once. Basically, there are many toxic mushrooms, poisonous mushrooms. It's not easy to differentiate a poisonous one from edible one. But we have a list of 2,100 species of mushrooms that are kind of listed as edible mushrooms. But it is important. It is good that it would be safe if you avoid eating wild mushrooms, even if they look similar to the existing known species. But because for many of these wild species, I mean, for most of the wild species of mushroom, there is no knowledge or scientific knowledge about the edibility. Fungi have been studied in botany because fungi cause, notorious diseases in plants. And probably plant pathology and botany, mycology became part of the botany. Not only in plant, fungi cause diseases in humans, for example, nail infection, vaginal candidiasis, ringworm, and candida infections of the mouth, throat, and esophagus. But not all fungi cause disease. I want to stress here that not all fungi are bad. Okay, like not all people are bad. Same way, not all fungi are bad actually. So many of the fungi collaborate with other life forms. For example, mycorrhizae. This is a beautiful relationship between fungi and plant root system that actually basically is founded based on the nutrient exchanges and communication. Okay, and fungi collaborate with lichens and cyanobacteria form. Fungi actually collaborate with algae and cyanobacteria to form lichens. It seems in this collaboration there could be as many as 3 fungal species. And as you know, lichens are known as they could be used as indicator of climate change. As you know, Fleming's penicillin producing strain is the Penicillium rubens. This is the phylogenetic tree which confirms that this is the Fleming's fungus, Penicillium rubens fungus in artificial medium. This is how the fungus looks. Penicillin is not the only one molecule we got from fungi. There are many which are being used as human medicines. Here is a list of FDA approved medicines from fungi. Last point, but not the least, fungi are saviors of this world. If Earth is ever hit by an asteroid, it seems only mushrooms can save us from going extinct. Please Google and find out why it is so actually. Okay. Now coming to the more technical aspect of it. So what is taxonomy? In an office setup, you have a records management system, filing system. You will not have one file for everybody or every department. Each department will the information about one department will be filed under filed in put in one file or one folder actually. Similarly, information about the biological diversity has to be managed very effectively in a simple manner, so that information is accessed in a very logical manner, and which can also be connected to the science of hypothesis testing, or scientific way of hypothesis testing. So here is the science here is that science. So that science is taxonomy. So taxonomy is a science which deals with classification of organisms, and it's not just based on morphological similarities. The taxonomy is based on deals with the classification of organisms based on the evolutionary traits. So we have imbibed Darwin's theories, Darwin's findings in taxonomy. So taxonomy has got three main components. The main component is classification, and the classification is hierarchical. So you start from basic say unit is the species, then you go up to the domain and life. Okay, then you have to identify this. If when you are in day-to-day life, you have to identify organisms. So as I told you, it is a binary process. You characterize an organism based on the characters based on the characters you identify. Use those characterization or those characters and compare with the known literature. Then you find out whether this is X fungus or it's Y fungus, or it is an unknown fungus. So once you know, even the fungus is already known, then it is bound to have a name or a taxonomic name. If there is no name, so if it is an unknown or novel fungus, then you have to give a name. That is a nomenclature. So common names can be given by anybody, but the scientific names follow strict rules of nomenclature. So going back to the history of a taxonomy, Carolus Linnaeus is the father of biological classification. So here is this great work, Systema Naturae. Going forward, the traditional classification of fungi included both mycelial and plasmodial fungi. It was a part of kingdom fungi. It was a subkingdom. And the subkingdom fungi had Myxomycota and Eumycota. And Eumycota had got these many, taxonomic categories: Mastigomycotina, Zygomycotina, Ascomycotina, Basidomycotina, Deuteromycotina. If you see many of the textbooks, you can still this classification, being presented as classification of fungi. This has to be updated, because in 1978, the life on the earth was divided for taxonomic purpose into three domains by the work of Carl Woese. So we have now bacteria, archaea, and eukarya. So it has been found that fungi are closer to animals than plants based on the molecular data. So here you see animals, here you see fungi, here you see plants. Basically, this clade is closer to animals. Therefore, fungi are closer to animals than to plants. So in 1990s, there was a series of papers on PCR primers for fungi. So one of the important book publications is this, actually, by White et al. I think it might have been cited at least more than 1.1 million or 1 lakh times actually. So this is an important paper: amplification and direct sequencing of fungal ribosomal RNA genes for phylogenetics. Here you have got very famous ITS1, ITS5, ITS2, ITS3. These primers are being used now. But now also we are using these primers. So this was published in 1991 or 1990. Okay. So this kind of work led to the generation of nucleotide data or the DNA sequence data for fungi. So the traditional morphology-based taxonomy started embracing molecular data, DNA sequence in the taxonomy. So that led to many changes in taxonomy. Also in 2007, the fungal classes of fungi were redefined phylogenetically. So now you have traditional zygomycota is not there, and traditional chytridomycota is used in a restricted sense. We have microsporidia as a part of fungi, So there are many taxa, So which sound very unfamiliar, but they are part of the modern classification of fungi, which is a monophyletic unit. So that was at the family. That was at the kingdom level. At the species level, there is a concept of using short fragment of DNA sequence data to identify organisms. That is called DNA barcoding. In case of that started with butterflies and animal kingdom, using cytochrome oxidase 1, and but cytochrome oxidase 1, COX1, was found to be ineffective in case of fungi. Therefore, as a consensus, ITS region was proposed as the DNA barcoding region for fungi. But this is okay for up to genus level. For many groups, this is not effective in species identification, especially in Collotrotrechum, that I am going to talk. It is important to know about the dual nomenclature in fungi. So, in a typical fungal life cycle, we'll have sexual reproduction and asexual reproduction. Okay, the sexual reproduction results in teleomorphs, and asexual reproduction leads in anamorphs or asexual structures. But sometimes a fungus may just produce the teleomorph, or sometimes fungi may just produce the asexual structures or anamorphs. What do we do with that actually? But in older times, fungi which produced only asexual structures were put under deuteromycotina. Okay, and the sex was considered very important in the classification system, sexual structures. So these were given more importance, and these become the anamorphs or asexual fungi become kind of orphans. So it was expected that a fungus should have information about, it should be associated with the fungal description should have both teleomorphic information and information on anamorphs. If the only anamorphic information on anamorphs is there, but the telomorph information is missing, then there was a problem. So this kind of fungi, anamorphs or sexual fungi, were treated not fairly. And it is very important to know why fungi produce this kind of, why fungi have this kind of pleomorphic life cycle. Having different kind of different kind of structures. Let me give an example. This is the, you have, ascomycetes, teleomorphic structure of a fungus, and the same fungus can produce anamorphs, asexual structures. So they look very different. Okay, in a traditional system, which is based on morphology, this would have, if the teleomorphic information is not supplemented with anamorph information, it would be published as a valid taxon given importance, but anamorphic taxa would be given less importance. It would be part of Deuteromycosina. But if a fungus is associated with, teleomorph, anamorph, non-sporulating state mycelia, so all this information is there, then it is known as a holomorph. But you cannot, in nature, the fungus may not produce teleomorph, anamorph, and non-sporulating state at the same time or at the same place. So that leads to problem, resulting in the dual classification system. One classification system for the sexual fungi, another system for asexual fungi, anamorphs. So there are three types of asexual fungi: hyphomycetes, coelomycetes, agonomycetes. Okay, because of this DNA sequence information, ability to sequence the ITS region and other gene regions. So now, then in 1919 onwards, taxonomy started sequencing ITS from these asexual fungi, the cultures of these asexual fungi, especially hyphomycetes, coelomycetes. Of course, they were sequencing teleomorphs, but when they were isolated, they were sequencing the ITS and other gene regions like 18S, 28. So they could connect these asexual fungi with the with their telomorphic counterparts, because in a phylogenetic tree you if you have molecular data, you can place both endomorph and teleomorph in the same tree. Basically, you can come up with a single classification system for both teleomorph and endomorphs. So but how do you know? I mean, traditionally, some morphological characters were treated as morphotaxonomy characters. It was assumed that these characters are very important in terms of evolution. For example, it was assumed. But in the context of molecular phylogenetics or the incorporation of DNA sequence data, how do we assess the phylogenetic importance of morphotaxonomy characters? So what we have been doing is, if you have a fungal culture, we actually extract the DNA from it, then do the PCR of ITS on other gene regions, then sequence those gene regions, and analyze using Mega and other software. When the phylogenetic tree comes, if the morphological character is phylogenetically important, probably it will result in a monophyletic clade or a single cluster of a species, or the taxa actually. This is how we started assessing the phylogenetic utilities of morphotaxonomy characters. Let me give an example from my PhD work. There are two genera. These are anamorphic genera. These are asexual fungi, Spadicoidetes and Diplococcium. The morphotaxonomic characters were catenation of conidia, if the conidia are in single or if they are found in chain, and the branching of the conidia force. So these were the two important morphotaxonomic characters to separate them. Okay, so when I was a PhD student, I somehow got into this project, and this became a part of my thesis. So I got the cultures of these, and I sequenced some gene regions, and when I constructed a phylogenetic tree, it was found that both the morphotaxonomy characters are not phylogenetically informative. So these kind of characters, morphotaxonomy characters, can be found throughout the tree of life. That means it's a very primitive character which cannot be used in phylogenetic, phylogenetic classification. So then I went on to do one more study on polyphyly of Sporidesmium complex. So these are similar looking asexual genera: Sporidesmium, Repatophragma, Stangivacia, Elisembia, Sporidesmilla lincusia. I got the cultures from Wemping, who wrote a big manuscript, or who wrote a big book on Sporidesmium complex. So when I did the molecular phylogenetic analysis, I found that these Sporidesmium morphologically similar anamorphic genera are polyphyletic. The question is, of course, we found that these genera are polyphyletic. How do we deal with it actually? So when I visited Professor C.V. Subramanian in Madras, University of Madras, in 2004 or 5 or 6, I'm not sure. I think 2007, I guess. Not very sure, because I published this paper in 2006. Probably I met him in 2006 or 7. Okay, maybe in 2009 when I joined IMTech Chandigarh. So I told this is the paper of Sporidesmium, because Professor CVS actually had worked on Sporidesmium. He had published many papers. So I showed the paper to him. He was very delighted to see the paper, and probably he said that I have seen the paper, but you should do and he said it's a good contribution. You should do more. I didn't realize what he meant. I mean, I didn't understand what he meant. You should do more. Basically, he meant that it's not just enough to demonstrate that these fungi are polyphyletic. You have to come up with a better classification system for this anamorphic fungi. Anyway, I could not continue with this group of fungi because I went on postdoc and joined IMTech Chandigarh. But later on, I met my PhD advisor. He said, yes, we wanted to continue your work on Sporidesmium, but we were not so lucky. But somehow I didn't follow it up much. I think recently, today morning, I was searching for it. I saw one order, Sporidesmialis, I think under the Sodarium mycetes. This particular order was described by Pedro Cruz et al. In 2018, based on Sporidesmium as a type. Probably this is the way to go forward. Not just describe the polyphyly. You have, for every new understanding, we have to improve the taxonomic structure. This is one of the ways to do is you have to give new name, taxonomic, we're following the proper taxonomic procedure. I think that's what Professor Pedro Cruz did when he described Sporidesmialis order in 2018, based on the Sporidesmium as the type of that particular, I think, order actually. I think that Sporidesmele has got only one family, one genus. Probably I don't know how many genus, but Sporidesmele is the type. So now, as I was telling, a fungi can produce both sexual, spores or sexual structures, teleomorphs, and asexual structures, asexual, spores like conidia. But sometimes you may not see the full picture of a fungus. It might just produce, as I told, only teleomorphic structures. Like you can make an ascomycota, asci, and ascospores, and other structures are not seen in nature. So, how in this perfect world, what is a fungal species? Now, how do we know what constitutes a species actually? So, conceptually, species are segments of evolutionary lineages that are evolved independently from another. Basically, a group of populations which have stopped exchanging genetic material with the other lineages. Okay, there is a genetic independence, there is evolutionary independence. That is important. But when we talk about morphology, can we do that actually? That's a concept, but in day-to-day life, the concept has to be used in a practical way. So here we have got what we call species recognition criteria. Species can be recognized based on morphology or DNA sequence data or a combination of gene sequence data. When we use morphology, that becomes morphological species recognition. It's the recognition of a species based on morphology. When you use gene sequence data, for example, ITS or 18S or 28S RNA, this is recognizing a species based on gene data. That is phylogenetic species recognition. Here, the concept of monophyly is very important. It is important that you prove the monophyly of group of organism before you confirm it as a species or any other taxon. So, but you see, one gene region can tell the history of that particular gene, probably not more than that. So it is important to use more than one gene regions. So when we use more than one gene regions in recognizing a fungal species, and these genes should be unlinked, so that becomes a genealogical concordance phylogenetic species recognition. This is recognizing a fungal species based on genealogically concordant genes. Genealogically concordant genes means these genes should have shared similar history or evolutionary history. As you know, morphological species recognition is based on morphological characters. In case of Coledotrechum gliosporides, if you see this kind of structures, for example, this kind of conidia and this kind of aprisoria, this kind of conidia with this kind of this much of size, you can describe it as a Coledotrechum gliosporides. But in case of Coledotrechum gliosporides, this particular fungal name was associated with more than 450 plant genera. I think 1,000 plant species were known to be affected by these fungal taxons, one or other way, as a pathogen or endophyte or as a probably sapro. But if you extract DNA from this culture and amplify ITS region, then you can describe this particular fungal species in a phylogenetic way. Here, in this case of Coledotrechum gliosporides, this one particular, this fungus was described long back in 1800 plus, 1850 or some that particular time. So finding herbarium material for a culture for this fungus was not easy, and they propose one of the herbarium material or cultures as the type, kind of a type equivalent to type. I'm explaining in simple manner. So here is an epitype for Coledotrechum gliosporides. Of course, there have been so many cultures which claim to be, Coledotrechum gliosporides, but when a set of mycologists came together, proposed one culture as an additional type or epitype of Gliosporides that changed the whole taxonomy of Coltotrechum gliosporides. This particular culture became the authentic culture or reference culture based on which the taxonomy of Coltotrechum gliosporides had to be revised or improved. Before telling that, why do we need to use more than one gene region? I have already told, because one gene region actually tells the history of that gene only. For better understanding of the species history of that particular group of individual, better to use multiple genes, multiple unlinked genes. Actually, it should actually indicate lack of genetic exchange beyond that clade, actually. See, going back to the Gliosporides, I was telling here, the Cannon et al. Designated one epitype. So that resulted in a better understanding. Previously, it was thought that Coltotrechum gliosporides was a common pathogen on tropical fruits. If any tropical fruit was affected by anthracnose or such kind of things, for example, coffee or mango, apple, chili, so they would give Coltotrechum gliosporides. But because of molecular data or designated epittype, it was found that Coltotrechum gliosporides belongs to this particular clade, and it is not monophyletic. So other clades had to be given new names. So new names were given, like for example, Coltotrechum siamens, Coltotrechum fructicola, and Coltotrechum asianum, Coltotrechum horgi, and Kahave, simonsi, see, these are the names for morphologically similar Coltotrechum gliosporides sensu lato. So this is the Coltotrechum gliosporides in the modern sense or in a strict sense. These are all the new names for morphologically similar species. Okay, basically it is the better understanding of the Coltotrechum gliosporides species complex. So it was established that long back that excuse me, Coltotrechum gliosporides is a species complex. So we need to actually clarify the species boundaries, since that in particular species complex. In this beautiful paper, Ware et al. Actually demonstrated that Coltotrechum gliosporides species complex is a set of 23 phylogenetic species. These are the names of the 23 phylogenetic species under one morphospecies Coltotrechum gliosporides. Gunjan was working on Coltotrechum gliosporides, and she got many Coltotrechum species. We identified them as Coltotrechum siamens. When we used APMAT gene marker, we found genetic diversity, and we also sequenced other gene regions. Then we found that because there were so much of genetic diversity, we started describing the novel clades as new species. Actually, we published a paper. We published a paper resolving the Coltotrechum siamens species complex. We've used the phrase Coltotrechum siamens species complex, and we described a couple of new species of Coltotrechum, which in the so-called siamens species complex. But in later on, Chai groups, Lai Chai's group from China, they used new methods, they use coalescent methods. When they use coalescent methods, which is a sophisticated way of analyzing a group of fungi, they found that Coltotrechum siamens is a single species. It is not a set of 7 species as described previously by us and other mycologists, actually. So this is based on the coalescent methods. What are coalescent methods? It's a deviation from the phylogenetic species recognition. Here, lineage sorting is involved, and incongruent genomic regions are considered when the phylogenetic estimation. Basically, the coalescent methods incorporate population genetics principles. It is not just the idea of genes sharing similar evolutionary history. There are some other gene regions which might be a little bit different. So we need to consider those gene regions, genomic regions, in the phylogenetic estimation. So this is about the coalescent methods. In case of Coltotrichum siamens, it was found to be a single species because there was no genetic exchange beyond this clade. Okay, and to prove that, they use the mating test. So these 7 species, what we described before, they could actually mate together and produce this sexual structures. If they were separate species, probably they wouldn't have produced the sexual structures, because the genetic barrier should have been there if they were different species. That is the idea. So here are some guidelines. I'll take 5 more minutes. I will tell 11:52 now. So if you are trying to describe a fungus based on morphological species recognition criteria, so it is important that you examine the holotype or the herbarium specimen matter, herbarium specimens actually. Okay, taxonomy is based on type materials. If your analysis doesn't include type material information or the sequences from type material, it almost inconsequential. Okay. It's better to consider whether it is the intraspecific variation or it's more than that. Sometimes the spore size may be just some of the spores may be larger by 3 micrometer or 4 micrometers. So are you going to describe it as a new species, or it is just a part of intraspecific variation? So that you have to make sure. And you have to always, as I was telling you, have to always examine the specimens. And if the taxonomy epitatypes, if the holotypes are not there, if you are a taxonomist, you can actually go for epitaphification, that is the designation of additional type materials, based on which you can improve the taxonomy. If you are focusing on DNA-based, species recognition, you cannot just take a couple of sequences and say it's a new species. You are expected to use a population, in the sense you are expected to use maybe more than 5, more than 10 fungal cultures and sequence them. I have seen many papers describing new species based on one culture and one sequence. So does one culture represent one population? It's quite probably unreasonable, and sometimes probably borderline irrational actually. Of course, as I told you, you cannot use just ITS to describe a new species. You are expected to do multi-locus approach or multi-gene region. And in addition to multi-gene, that means genealogical concordance, phylogenetic species recognition, also go for coalescent methods. Okay, so species boundaries, as I told you, you can use the genealogical concordance, phylogenetic species recognition, coalescent methods. But in future, it is going to improve like anything. You will have a very you will have more refined way of describing or delimiting species boundaries. It would have molecular data, morphological data, ecological data. We would be using. It will be leading to integrative taxonomy, and basically it will focus on eco-evolutionary theory, which involve designing experiments, contextualizing findings in broader co-evolutionary process, and improve the prediction of species patterns. So future is going to be exciting, but to do better, to embrace future, we have to do good in present. So I hope this understanding will help you to embrace future in a more exciting manner, more comfortable manner, okay, without much resistance. Thank you very much, Dr. Rohit Sharma, for this opportunity. Thank you, ladies and gentlemen, for patiently listening to me. I would like to acknowledge the support of my organization, CSIR-NIO, and also acknowledge CSIR Imtech Chandigarh, Kunjan Sharma, and Yamini, Vasha Shinde. These are my PhD students, Genevieve, my collaborators, Kevin Hyde, Professor Kevin Hyde, Bevan Ware, Laicha, and many more. And I want to acknowledge, I want to declare that many of these slides, many of the photos are from the canva.com Pro version. Ladies and gentlemen, if you have any questions, please do ask me. I'll be happy to answer if I know the answer. Thank you.
Yeah. Uh, Thank you very much, Dr. Shenoy. Thank you very much for the talk, excellent talk. I'll just take a couple of moments before we go into question and answer session to thank our senior mycologist, Dr. Bhatt and Dr. Sridhar. They were kind enough to join for the talk and make it a success. Coming to the question sessions, I think there are three persons who have raised their hands. Dr. Vidyanath Jha. Please go ahead, sir. Dr. Vidyanath Jha, please go ahead with your question, sir. Okay. Dr. Abhijit Singh. Mr. Abhijit Singh, can you go ahead with your question? Okay. Mr. Raghavendra Rao. Okay. Mr. Rajshri Rit.
Okay, I have unmuted. You can go back to the list again, Rohit. Mr.
Okay,
Jha,
okay.
Dr. Jha, please.
Dr. Jha, please go ahead with your question, sir.
It was by mistake. I do not have any question. Thank you for your presentation.
Thank you.
Mr. Abhijit Singh. Okay. Mr. Raghavendra Rao. Okay. Mr. Rajshri Rit.
Hello, I have basically three questions. First of all, I want to know, in order to define a genus or species, what percentage of similarity is required to define a species, and what percentage of similarity is required to define a genus? Is there any such values?
I don't believe in such values. Those are rubbish.
Okay, thank you, sir. And another thing is that there are several other methods such as FAMES analysis and immunochemistry based identification, and does they play some significance in classification and identification?
In some groups like Xylarialis. Okay, and
Okay, thank you, sir.
Aspergillus penicillium. Yes.
Thank you, sir. And my third question is when to go for 18S RDNA sequencing and when to go for ITS sequencing?
ITS sequencing. ITS sequence is the first step. Okay, and you get some idea. Then you decide whether to go for multi-gene, and if you want to go for multi-gene, you refer to the recent papers and then decide which genes to sequence and which methods to employ, whether genealogical concordance or coalescent or more than that, or whole genome. Because some of the groups have already embraced whole genome sequence data. Okay.
Thank you very much, sir.
Thank you.
So there is one question. Hand has been raised by a name T. I cannot see his or her full name. Do you have a question?
Yes, sir. I do have a question, sir.
Yeah. Please go ahead.
This is Tunduk Dolma, sir. Sir, basically, I have three questions. My first question is, sir, if we identify the fungus on the basis of morphology, so, sir, there is any key has to be followed for the same, sir? Which key has to be followed, sir? Because it is very difficult to identify on the morphology, sir. Because I am facing a problem in this.
You can use the keys which are published probably in 1990s, 2000. You'll get probably you can go to genus level or maybe family level, but after that, because the species description has changed for many groups actually. Species boundaries have changed. So these morphology based keys probably will have will not have so much of value as you believe in them actually. No, the value of those keys have reduced, but we don't have any replacement for that. We don't have replacement keys which can incorporate both morphology and plus polyphasic data. We are in a little bit mess. So the reliance on taxonomic keys are not much. It cannot be very much helpful actually in many cases.
Okay, sir. Okay, sir. Thank you, sir. Sir, my other question is, sir, as you said that ITS region amplification of only ITS region will not give you a species, will not give it as a species level identification, sir. Then, which region has to be amplified for the species level identification, sir?
That's a good question. I mean, very commonly, frequently asked question. When you have, if you have ITS sequence data, you compare
Yes.
with this type sequences available in GenBank or MycoBank, okay? Then you will get a fair idea to which group it belongs to. So, for example, if it belongs to Penicillium, you go to the recently published new species in Penicillium. That people
Okay,
that paper should be having the latest information, the state of the art kind of information, whether to use
Okay, sir.
morphology, whether to use more than morphology, if it is multi-gene, what genes. You should look at the gene regions they have used, whether they are used only genealogical concordance, phylogenetic species recognition, whether they went for, whether they opted for coalescent methods or did they try something new. So basically, based on ITS, you go to that particular genus in index fungorum or MycoBank, go to the recent paper, recent paper on the new species in the group. That should be your starting for you.
Okay, thank you, sir. Thank you very much, sir. Thank you very much, sir. It was a nice lecture. Thank you, sir. Thank you very much.
Thank you for joining.
Thank you, madam.
Thank
Thank
you
you
very
for joining
Thank-
as well as asking your query. Dr. Shenoy, there are some questions listed
Thank
on the chat box. I'll go one by one. Mr. Prasad and Ms. Ashwini has relatively similar questions. What is monophyletic and polyphyletic?
I mean, strictly speaking, a monophyletic group is a group of organisms or sequences which have hypothetically originated from the same recent common ancestor. That is the definition of monophyly. The polyphyly is that for a group of organisms which may look similar, for example, Sporidesmium, and similar genera. But when you do the phylogenetic analysis, you find them clustered all over the tree of life of fungi actually. So that means these may look similar morphologically, but they belong to the diverse phylogenetic lineages. That is the kind of explanation for polyphyly. Poly is multiple, phyly is origin actually.
Thank you, Dr. Shenoy. So the next is of Mr. Akshay. He wants to know that nowadays molecular sequencing is compulsory for recording a new fungal species or a new record to an area in case of macrofungi. Means he wants to know whether while publishing a new record also ITS sequencing is compulsory or not.
See, nobody is going to force you to sequence and force you to sequence and present. It is expected that when you propose something as a new record, you give the best possible data to the paper in the manuscripts. So if the reviewers accept your proposal, that's fine. So there is no need to go for a new sequenced, gene sequence data for new record. But if the reviewers say, this new record might require some additional information in the name of in the form of a DNA sequence data, they would suggest. It's up to you. If you don't agree with them, you go to another journal. Okay. If another journal rejects, do not accept your proposal of new record, then you go to the third journal. So when 3 journals say this new record needs to be supplemented with additional information, that means there is something in that. So that means your proposal needs to be strengthened with additional data. So it would be better you sequence that particular fungus and provide the information. I mean, more or less nowadays, the new species of fungi are to be supplemented with, or actually it has kind of become mandatory. There is no rule, okay? Nobody has framed a rule that every species, new novel species recognition, species description should be, supplemented or provided with the DNA information. See, but if you don't give DNA information, you are not going to integrate your beautiful fungus to the tree of life. So that's kind of incomplete contribution to the taxonomy. I mean, but I know many places you may not have resources to describe. So in many cases, many of the so-called new species couldn't be published because of lack of funds or inability to understand the newer concepts in molecular biology. So, but sooner or later, we need to upgrade ourselves. We cannot say we don't have funds, we don't have expertise. If the funds are not there, you have to collaborate. If the expertise is not there, you have to develop the expertise, or at least common understanding of it. If you do not want to get that expertise, then collaborate with the experts like Dr. Rohit Sharma.
Thank you, Dr. Shenoy.
Excuse me, sir. Sir, can I?
Yes, Aru sir, go ahead.
Okay, thank you so much, sir. So first of all, it was very an informative lecture. Thank you so much, sir, for this lecture. Sir, I just have a query that many a times I have seen that we describe a new taxon only on the basis of molecular details. So I just want to ask, especially in case of endophytic fungi, I have seen where we know that in many cases the sterile fungi are there. They are not producing any morphological characters. So although morphology is a very important character while describing a new taxon, but I have seen many papers where the mycologists, they are describing new species and they are just excluding the morphological characters. So I just want to know up to what extent we can do this, and up to what extent we can say that it is authentic, and where we can do this. Thank you so much, sir.
Actually, nobody is excluding morphology, because of these fungi are not producing morphological, morphotaxonomic characters like in other group of fungi in artificial media that we have developed so far. Suppose if you develop an artificial media which is suitable that for fungus, and that in that particular new artificial medium, if the fungus shows some beautiful morphotaxonomic characters, that would be wonderful contribution to taxonomy. But we do not have patience and resources or expertise to come up with new artificial medium, designing new artificial. What we do is we just report that in these PDA or MEA we couldn't see the morphotaxonomic characters, or not very vibrant morphological characters. So we report that, and then we rely our species proposal or new species proposal based on DNA sequence data. So it is my argument is that if new species could be described solely based on morphology till 1990, why there should be a problem of describing new species based on just molecular data when the morphological data cannot be kind of induced or cannot be seen very easily. So, but what I see in future, morphology would be kind of supplementary. Molecular data would be primary. So the role change would happen actually. Whether you like it or not, it will happen.
Okay, sir. Thank you so much.
Dr. Rohit,
Yes,
a couple of points. You see, morphology and molecular, characters. These two are essential.
So you are muted, sir. Please unmute yourselves, sir.
Okay. Yeah. See, morphology and molecular basis. This molecular taxonomy. This is the present scenario we have. But the question what she asked now is whether we should use, why we are using only molecular characters. Because in some genre like musk order, there is no morphological features essentially in endophytes. So they are using only the molecular features. That's fine. But I'm seeing some other scenarios. See, some genera like Fusarium, Aspergillus, Penicillium, Collotrichum, all these, there are numerous strains and the differences are very minor. If you look at Fusarium alone as a genus, right from the time of Wallenweber and Rinking, that's 1937, they wrote the monograph on Fusarium. Then came Snyder and Hansen, monograph. Then came, by Colin Booth. But all through these hundreds of years, last 100 years, we don't have a static classification even for Fusarium. Yeah, just because there are so much of differences within the species, even amongst the species. So in such places, the molecular basis would be very fine. But if you go to the Dothidiales or Solidariales, there are genera where you can distinguish them merely by morphology. So morphology still has a lot of value, but in future, it's always essential to have, as Dr. Shenoy said, if you give the molecular basis for that, then that is the permanent feature based on which we can do the identification in future. But for all the beginners, I would advise them that morphology still has a lot of value. Take the fungus, look under the microscope, see what are the features. I think we all did this in our early stages, and that has a lot of value even today. You should do that in future also. This is my advice to them. And it's good to know everything about fungi, as I say. Nice talk, Damodar. I enjoyed listening to you. Okay.
Thank you, Professor. But he is the guru of my hypomycete taxonomy. He taught me my hypomycete taxonomy to me in Hong Kong airport when I was about to go back to India. I hope you remember that, Professor. But okay,
yeah, I remember.
I'm not undermining the role of morphology, but what I'm saying is that we have to be kind of neutral towards all kind of data. So if you can generate diverse kind of polyphasic data, you should have a mind of reason and logic. And if Dr. Bhatt was telling some groups, you could differentiate two different genera based on morphology, fine. One morphotaxonomic characters, a set of morphotaxonomic characters. But if you believe in species as a hypothesis, you need to ask yourself, whether those taxonomic characters are phylogenetically informative. So there is two school of thinking here, So finally, we have to find a common ground. Professor Bhatt said that some group you can differentiate based on beautiful morphological characters. Of course, they are beautiful. They could be practically used in day-to-day life to separate them into two genera. But when if a person who is good in phylogenetic analysis, I'm not talking about myself, there's a set of people who have been employing this kind of sophisticated phylogenetic analysis, they would like to know whether all these morphotaxonomic characters you have been using to separate fungi into genera, are they phylogenetically informative? Whether these characters were really important in separation of the fungi into two groups, or two species, or two genera. But this questioning will continue. But in day-to-day practical things, you could always start with what you see.
Yes.
Yeah. But if seeing is kind of believing, not fully believing, but then you have to go for once you believe a little bit, then you have to probe further and actually believe using molecular data. In recent times, coalescent methods.
Yes.
What happened? Coalescent methods. There was rampant description of new species based on multi-gene sequence data without bothering about species boundaries. When the genealogical concordance, so when the coalescent methods have been started using in Collotrichum, Siamens, I think recently in Aspergillus. So now there is a rethink. Did we actually rush into describing new species. It just based on a set of molecular data or set of genes. Yeah. So there is a kind of caution. No, there's a थोड़ा आगे जाने से पहले एक step पीछे लेना है. That's the approach.
Correct. Yes.
Yeah, this thinking is coming from the legends of my fungal taxonomy actually. So they are thinking, did we kind of haste? Were we in haste when we describe all these new species and new taxa based on morphology plus multi-gene data, without asking ourselves whether actually they represent a set of a group of individuals or a population which was stopped, interbreeding, So here the thinking of population genetics, comes into play. So basically we are improving ourselves and refining our thinking process.
Thank you.
Yeah,
Thank you.
yeah, thank you.
Thank you very much, sir, for the nice discussion, because these discussions will help the youngsters to clarify some of the doubts that they are having in mind, and that these can clarify. I'll go with Dr. Anita Tiwari. Please go ahead with your question. She has raised your hand. Yes, ma'am.
Good afternoon, sir. Just 1 minute, please.
Good afternoon.
Sir, just 1 minute, please. मेरा वो बंद होने वाला. जी. Okay, sir. Actually, my question is that we are doing barcoding of fungus. So in barcoding of fungus, we are using 18S, but it is ITS1 to ITS4 region. So which region is basically significant for barcoding of this macrofungi?
Madam, personally, I don't think the concept of barcoding is losing a sheen. Okay. Especially in the case of fungi.
Ji.
Because even the ITS has got limited value for me in many groups,
Ji. Yes,
and though we started in a single locus, one gene region as a barcode region, this is it's an oversimplification actually. आपको लगता है कि हमारा आधार कार्ड सिर्फ हमारे एक फिंगरप्रिंट से हो सकता है? नहीं, there would be many other data, so that would identify actually. So,
sir, actually my question is just because some specific region of Madhya Pradesh is having specific type of fungi, so that is which is not available anywhere. So how come we make them to some code? How we can give code to that fungus? Like our Bandhavgarh part is having some different, very beautiful colored, which I have not seen even in the Google. So how come?
Let me answer. I mean.
Yes, sir. Yes, sir.
I would request you to go back to the recording of this, or maybe when it will be on YouTube.
Okay, sir.
Because I have explained that ITS as a barcode region is almost losing its sheen. Okay, ITS is one of the many markers that could be used, and you could use ITS as a starting point. In your case, to identify, I'm assuming that you already know the morphology of your fungi or mushrooms from Madhya Pradesh. Okay, and if you are not come to some conclusions based on of its novelty as new records and new species, then you start with the ITS sequencing. Then, as I told one of the members here, you go to the recent papers and recently published new species in that group. That should be giving you all the ideas how to describe it as a new record or a new species. So barcoding probably is now less talked about actually, because I was a part of DNA barcoding project in Canada.
Okay, sir.
Yes, thank you, Dr. Shah. So I'll be going some questions on the chat box that Ms. Pushpa asked that how can we compare the marine microscopic fungi with the terrestrial fungi based on morphology?
And actually, there is I think, as I was explaining to somebody in, can you hear me?
Yes.
See, I think Devadatta is the expert in marine fungi, but if you see strictly, the members of Halosporiales are the strict marine fungi. So there are some 100 species of halosporiums. Those are strictly marine fungi. You cannot find them in terrestrial environment. Okay. So answer to your question, you will not find strictly marine fungi in any other ecosystem.
So the next is Ms. Ashwini. Can one fungus, one species, change into other species after multiple subculturing?
Do you think fungi can evolve in 100 days? A genome size of 30-40 MB, right, can change in for 30-40 days or 1, 2, 3 months. I'm not sure. You need to test it.
So, so is there any scientific method to differentiate between edible and non-edible mushrooms?
No, sir, there is no method. You should talk to Professor Samantha Karunaratna, who is an expert in mushroom identification. Maybe Arun Kumar Datta, maybe Dr. Pradeep Kumar, maybe Arun Kumar from Gurayur, that college. So they would definitely agree with me that there is no standard method of identifying chemically or based on DNA whether one particular mushroom is edible or not. It's based on the trial and error method, traditional knowledge, and which have been documented in various scientific literature. Okay, but edibility is based on trial and error.
So I am skipping some of the questions because it has been already answered in other questions. So Fatima has asked that is morphology-based taxonomy of fungi becoming outdated? I think Dr. Bhatt has put a light on it that it's not still outdated. It still needs to be taken hand in hand with the phylogenetic one. So,
I want to, Rohit,
yeah,
I want to just respond to it. It's nothing called outdated. You need to embrace all kinds of data.
Yeah.
So we have a rich tradition of morphology of 150 years from Carolus Linnaeus. But the newer forms of data coming. In case of DNA sequence data, probably we'll have the whole genome sequence data, maybe biochemical data or ecological data. So we need to be open-minded, just not say this is going to be outdated or not. We have to have a very critical mind. So whether the new species or new taxon I'm describing based on a set of data is good enough, rather than saying the morphology is outdated, getting outdated. There is no need to, no need to be a little bit upset that the morphology is going updated, or outdated. After some time, multigene is also going to be kind of outdated actually. So in taxonomy, so many new species, what people described have become obsolete, invalid. So taxonomy is very vibrant and it can have a very humbling effect. For example, we described a couple of 3 or 4 new species. I think 2 species in Siamens. So those species are not acceptable now. They are merged with Siamens now. For example, the popular Coleotrechum capsicum is not capsicum; it's a Coleotrechum truncatum. So there is no permanent nature of taxonomy. The taxonomy is always changing. So in that method, morphology is going to be embraced, integrated, case to case. Okay, sometimes it will be given more importance, sometimes it will be given not so much important, based on the way we look into the things.
Thank you, Dr. Shenoy. So another question is by Anusha. She wants that if you can elaborate on powdery mildew classification and taxonomy, especially anamorphic characteristics.
Sorry, I am not an expert in powdery mildew. I think people from Gorakhpur are expert in that. I don't know, who would you suggest, Rohit?
I think Professor Kamal, or we can have some persons to have a special talk on this powdery mildew. We can have lined up in some of the talk in coming days.
Maybe Dr. P.M. Singh, probably.
Yeah, P.N. Singh or S.K. Singh. So then Dr. Singh has asked that how to identify a fungus, including using the modern tools and technique to reach to the species level identification using multi-gene sequencing.
I think I have already answered. You start with ideas. Once you get some idea, go to the recently published paper on new species in that group. That should give you some ideas to go forward.
I think most of the questions we have taken. If something else is, there is one hand raised, I think, by Mr. Anand. Yes, please go ahead, Mr. Anand.
Hello, can you hear me?
Yes, sir. Yes, sir.
Dr. Damu, it is a nice talk. So my question is related to one of the question recently asked about after several culturing, whether that fungi is going to change the species. So this is my personal experience when my students sequenced, like ITS sequenced, and a few months back it come to some species, and later on same isolate extracted and sent for sequencing just for check. It has comes under different group, different species. So my concern is, see now latest by many people, what they do, they will do one analysis. Once they will ITS sequence, they will do and just align and they find one species related to that, and they will say that this is my species, and this is going to publish, as well as they are going to enter in the gene data bank. So if this is a wrong, so what is going to happen in future? If some of these are not, like as you said, we have to use multiple factors to identify a species. If somebody is done in only using ITS and they enter and they keep that in database, so in future, whoever search for that sequence, it will come as that species. And what is the, I don't know, that is, if anything can be
Okay, Dr.
answered.
Anand, I got your sentiments actually. Number one,
Yeah.
so your a personal experience of one ITS becoming another ITS sequence, different groups. So I would definitely look into in this following ways. Assuming that the culture is pure, there is also a possibility of multiple copies of ITS in a fungal genome. So there is a report of that in one of the reports I was reading. Please go through that publications. Maybe we'll read together and discuss. Number two, ITS cannot be the only way to just to tell that it could be a new species. So I would suggest go for multi-gene sequence data and define the species boundary based on coalescent methods. That should give whether it is a new species or it is a just artifact.
Yeah, I may not go for the final decision by that, but people are doing that. That is why my concern. Like
See,
many groups.
when we started, when I started my PhD in 2003, there were people who would submit anything to GenBank with any name and any quality. That is still a concern. But what we have to do is there are sequences in MycoBank, and you have to rely on sequences from the type materials, and unfortunately, many of the type materials are eaten by rodents or misplaced by the taxonomists. So there is no single place you can find herbarium specimens, or for that matter, all the cultures of new species or new taxa. At least in bacterial taxonomy, it is expected that you deposit these cultures or specimen associated with new taxa in two international culture collection. Here, the fungal taxonomy is very chaotic that way. So, but we have to work within the limitations, and progress has to be made within the limitations only. We cannot go beyond that because we are individuals.
Okay, thank you, doctor.
I just said I agree with Dr. Shenoy that there can be multiple copies of ITS. So even if you sequence of the same culture again, even without subculturing, there may be some variation in the sequences also because of some other kind of gene sequence. And I think some people also do SSR kind of thing also, that repeat sequencing, short repeat sequences to start this 10 level differentiation. So that also one can go. And so there is a couple of questions more, Dr. Shenoy, that is there any app to identify fungi?
Talk about fungi, we are talking about 2 lakh fungal species, which in various forms, mushrooms, molds, yeast. So to have one app dealing with so many diverse fungi probably is impossible at the moment. Maybe after 10-15 years when I'm retiring from NIO, probably I would see that.
Yes. So there is one more question, like Dr. Tiwari, what is the concept behind one fungus, one name?
Basically, you cannot have two names for same fungus. Okay, it used to be given. Apparently, we cannot prove that these were given. Because the idea situation was very imperfect, not conducive to find out all the life forms of one fungus together in one place. So in that situation, imperfect situation, there was an imperfect taxonomic system or classification system of describing endomorphs under Deuteromycota, and endomorphs with or without endomorphic information endomorphs as a main system of classification. But because you see, one fungus even it produces multiple forms like endomorphs, telemorphs, non-sporulating, but the DNA is the same. In somehow, if you get lucky, you can and get all the forms in kind of in one place or somewhere by the chance. You could always sequence the ITs from all three different forms, like butterfly. It has got larva, pupa, and adult. So they are not different species, right? They are the same species. So if you extract DNA from all the different stages of a butterfly, it should give you the same cytochrome oxidase gene, right? Similar in fungi. Because of modern DNA sequence data, you could now make attempts to integrate the dual system into single system, but that is in the process. Everybody has to work for it. It's imperfect, it's chaotic, sometimes quite frustrating also. But the idea is to have one classification system for all fungi.
And there is an important question, Dr. Shenoy, for the youngster, especially Mr. Ahmed has asked, how can we select an outgroup while making a phylogenetic tree?
If you're studying about humans, you make butterflies as outgroup. If you're studying about humans and butterflies, you make probably fungi as an outgroup. So if you're studying as Coleotrichum gliosporidis, probably you make the Boninense as the outgroup actually. So if you should define what you're focusing on. That is an in-group. If you're well aware of in-group, then you know which one is outgroup.
I think most of the questions and all the questions are answered, because there is one question left, that is how we can identify fungal culture which has several and doesn't show any sporulation. So I think probably we have discussed this question earlier as Arsu has asked. So I think we'll end this. Shall we end this question answer session, Dr. Shenoy? I think.
Yeah, you could share the feedback form if you have.
Sure. I'll share the feedback form over
and it will be open for half an hour. Then if anybody interested, they can give us a feedback till 1 o'clock.
Sure. I have shared the feedback form. Please give your feedbacks on it. I think the link is visible to all, and please you can go ahead with it. There is one question of Mr. Prasad. How far will haplotype analysis help in the establishment of a new species?
I'm not sure about, probably in some groups actually, Aspergillus and all. I mean, if you're talking on holotype, probably yes. Haplotype, I'm not very sure.
Yeah. Thank you, Dr. Shenoy, for taking up the question answer session. I think we all should give a big round of applause to Dr. Shenoy for taking all the questions for more than 45 minutes. And I think this series will be in continuation. This is lecture 1 series. Next month, it will be by Dr. Atri on mushrooms. And thereafter, in December, it will be Dr. Dagar on anaerobic fungi. So this is a series of lecture series that we have started. That will be on the every first Saturdays of every month. So please keep the dates and remember to join in. So I think I'll once again thank Dr. Shenoy for taking up the time for this talk. And I think this talk will also be, recording of this will be available over MycoAsia as well as the YouTube channel of MycoAsia. So those who have missed it, you can go visit that thing.
Yeah, so Rajeshri was talking about the feedback form issue. Rajeshri, it is sorted now. Okay, you can submit your feedback. Thank you. Responding to Dr. Rohit, thank you very much for your generous words at the beginning, and for being a wonderful host today and managing the whole show show in a very awesome manner. I think we had got really quality discussions today because of the
Yes.
presence of our great mycologists around,
Yes.
and some of the very enthusiastic participants asking some pertinent questions, very interesting questions, some basic questions, trying to show, I mean, getting into the groove of a complicated field of, fungal taxonomy. I must, let me be blunt, let me be blunt. Fungal taxonomy is not everybody's cup of tea, but there are very few fungal taxonomies. There is less competition. So you just have common sense. You would have more opportunities when there is a field is not very crowded. So just do consider that. Thank you.
Thank you. Thank you. And for any questions, you can join the MycoAsia group, those who are new. So there also you can post your questions. We have very good discussions over the WhatsApp group also.
I think we can just say bye to all of us. Right?
Thank you. Bye. Good day. Good day. See you all. Thank you.
Thank you so much. Yeah. Bye.
Thank you very much, sir. Thank you. Thank you.